All right, welcome back to The Explainer. Today, we're not just talking about metabolism. Nope. We are going on an energy heist deep inside your very own cells.
I mean, have you ever really thought about it? How does a sandwich actually power a thought? It kind of seems like magic, doesn't it? But really, it's this incredibly high-stakes, microscopic process. It's all about converting the food you eat into the fuel your body runs on, literally one molecule at a time. So our target for this heist is a single unassuming sugar molecule, glucose. And the prize, the big score, it's ATP, that's adenosine triphosphate. You've got to think of it as the universal energy currency that powers, and I mean literally, everything you do, from blinking your eyes to thinking your next thought. Okay, so every good heist needs a blueprint, right? Well, this is ours. This is the master plan, the overall chemical reaction for what we call cellular respiration. It looks complicated, but it's simple. We take one glucose molecule, mix it with 6 oxygen molecules, and through a bunch of really clever steps, we release a ton of energy as ATP. The only evidence left behind? A little carbon dioxide and water. So let's break down exactly how this job gets done. Our whole operation unfolds in 4 main stages. Think of each one as a distinct part of the plan, all designed to systematically strip the high-energy electrons from that glucose molecule and, cash them in for ATP.
Now, let's be clear. The whole point of this entire intricate process is just to make ATP. And our cells, they've got 2 main ways of getting paid, 2 different methods for creating this all-important energy currency.
All right, so first up, we've got something called substrate-level phosphorylation. The best way to think about this is like a petty cash grab. It's super fast, super direct. An enzyme literally just grabs a phosphate and slaps it onto an ADP molecule to make ATP. Boom, done. This is great for a quick burst of energy, like sprinting, but the payout, it's pretty small. Now, on the other hand, you've got the main event, oxidative phosphorylation. This isn't petty cash. This is the big, complex, planned-out vault break. It's way more involved, but the payoff is massive. We're talking a treasure trove of ATP. This is the sustained energy that lets a marathon runner keep going for hours.
So, how do we move all this stolen energy around from one stage to the next? Well, for that, we need getaway cars. And in our cellular heist, these are special molecules, coenzymes called NAD+ and FAD. Their job is to pick up those high-energy electrons, which turns them into NADH and FADH2. Think of them as armored trucks, shuttling that valuable cargo to the final payout stage.
Okay, you ready? Let's dive into stage 1, glycolysis. This is the break-in. It's the first step, and it actually happens right out in the open. Well, in the cell's cytoplasm anyway. No special keycard or security clearance needed for this part. So here's the rundown on glycolysis. You gotta spend money to make money, right? The cell actually invests 2 ATP molecules to kind of break that 6-carbon glucose in half. What you get are 2 smaller 3-carbon molecules called pyruvate. But don't worry, that investment pays off right away. We get a gross payout of 4 ATP, and we load up our first 2 getaway cars, making 2 NADH. So, when you do the math, we walk away with a net profit of 2 ATP and 2 NADH. See? It's that quick, direct, substrate-level phosphorylation we were just talking about.
Okay, so now we've got these 2 pyruvate molecules. They're now smuggled into the high-security zone of the cell, the mitochondrion. You've probably heard it called the powerhouse of the cell, and for good reason. This is where the real action is about to go down.
But wait, before we get to the main event, each of those pyruvate molecules needs a little bit of prep work. A carbon atom gets stripped off and released as carbon dioxide. That's kind of the first evidence of our heist being left at the seam. What's left is a 2-carbon molecule called acetyl-CoA. And importantly, during this little step, we load up 2 more NADH getaway cars, 1 for each of those original pyruvates.
All right, now that acetyl-CoA is ready to enter the citric acid cycle. You might also know it as the Krebs cycle. This thing is a brilliant metabolic loop that's going to completely dismantle what's left of our glucose molecule. And since we started with 2 acetyl-CoA, this cycle runs twice. As it turns, it releases the last of the carbon atoms as CO2, it generates a tiny little bit more ATP directly, but, and this is the most important part, it loads up a massive fleet of getaway cars. We're talking 6 NADH, and 2 of our other carriers, FADH2. They basically do the same job, but just think of NADH as carrying slightly more valuable cargo. That's going to matter a lot when we get to the final payout.
And now it's time for the climax of our heist. This is it. This is where all those fully loaded getaway cars, every single NADH and FADH2 we've collected so far, they all converge on the inner mitochondrial membrane. It's time to cash in their high-energy cargo for the grand prize. I mean, just look at our haul so far. At this point, our heist has been wildly successful. We have collected a grand total of 10 NADH and 2 FADH2 getaway cars, and every single one is packed to the brim with high-energy electrons that we stripped from that one original glucose molecule.
So what happens now? These getaway cars pull up to something called the electron transport chain, and they drop off their precious cargo, the electrons. As these electrons get passed down a line of proteins, kind of like a bucket brigade, they release energy. And that energy is used to pump protons across a membrane, building up this really powerful gradient. But here's the absolute crucial part, the catch. This whole beautiful system will grind to a halt if there's nowhere for those electrons to go at the end of the line. They need a final destination, and that final electron acceptor is oxygen. Oxygen is the cleanup crew. It swoops in, grabs the used-up electrons, and keeps the whole assembly line moving.
And here is the payoff. That powerful proton gradient we just built, it gets harnessed by this incredible molecular machine called ATP synthase. The best analogy is a microscopic water wheel or a turbine. As those protons rush back through it, the whole thing spins, and with every single turn, it just churns out ATP after ATP after ATP. We're talking about the vast majority of our profit here, roughly 28 molecules. This is that massive, super efficient payout. This is oxidative phosphorylation.
So let's tally up the final score. When you add the couple of ATPs we made in the early stages to the massive payout from this grand finale, one single molecule of glucose gives us a grand total of about 32 ATP molecules. That is a phenomenal energy return on investment. And it's exactly why you can go for a long hike or stay focused during a really long day.
But what happens if our cleanup crew, oxygen, doesn't show up for its shift? What if you're exercising so hard you can't breathe fast enough? Well, without that final electron acceptor, the whole high-tech electron transport chain just backs up. It's like a traffic jam. It grinds to a complete halt, and now our getaway cars have nowhere to unload their cargo.
So, when oxygen isn't around, the cell has to switch to plan B. It resorts to a backup plan called fermentation. Now, you might be thinking, oh, fermentation, that's just to make a tiny bit of extra energy. But its real purpose is actually way more clever than that. See, the primary goal of fermentation isn't really to produce ATP at all. I mean, it doesn't make any new ATP beyond what we already got from glycolysis. Its real job is to solve that full getaway car problem. It takes the NADH, which is full of electrons, and basically forces it to dump those electrons onto another organic molecule, like pyruvate. And in doing that, it recycles the NADH back into an empty NAD+. So why is this so important? Why go to all that trouble? Well, because by freeing up those NAD+ getaway cars, it allows the very first stage of our heist, glycolysis, to keep on running. This is exactly what's happening in your muscles when you're sprinting full out and they can't get oxygen fast enough. That's what leads to the buildup of lactic acid. It's a quick, it's a dirty, but it is an absolutely life-saving solution to keep at least a tiny trickle of ATP flowing. Okay, so let's zoom out for a second and look at the whole operation. We just followed a single sugar molecule through this really complex four-stage heist, all to extract its energy. So what's the big takeaway here? What really matters? When you boil it all down, this entire intricate process, this beautiful cellular heist, it's really about one simple, but incredibly profound transformation. It's about converting the potential chemical energy that's locked away in the bonds of your food into the immediately usable kinetic energy of ATP that powers every single moment of your life.
And that leaves us with one last kind of mind-blowing thought. That CO2 that your cells release as waste, that's the exact same molecule that plants breathe in for photosynthesis to make glucose in the first place. So this tiny, invisible process that's happening in your body trillions of times a second, it's actually the other half of a grand planetary cycle of energy. It's a partnership that makes all life as we know it on Earth possible.